the quality assurance. And I congratulate the team for running faculty development program in a big way for the seventh day in a sequence. Madam, thanks for your introduction, but you missed one very key element of my introduction. Can you identify what it is? And it is very relevant to the topic.
Experience in
You mean experience in clinical trials?
yes, sir.
Yeah, in Institute of Clinical Research India, I was there for 4 years.
Yes, sir.
No, I was referring to, I am the chairperson of Institutional Quality Assurance Cell. So this has direct relevance with the today's topic, quality assurance in clinical trials, a regulatory perspective.
Sir.
So good morning, Dr. Mamata Madam, Principal, and HODs and colleagues, and my ex-colleagues from other pharmacy colleges, and the faculty fraternity at large. I welcome you all for a 1 hour session on quality assurance in clinical trials, a regulatory perspective. So as an example, I will be quoting always quality assurance in education, a Pharmacy Council of India perspective. So as faculty members, everyone should have a lot of intro into quality assurance in academics. So that I will be taking as an example throughout the session on each slide. So disclaimer is, it doesn't have any relevance to any college or any faculty in particular. But what pharmacy profession regulations say, we say in this particular session. It's all about the culture of training, monitoring, auditing, checking to ensure the quality assurance, without which, whether it is clinical trials or academics, is incomplete. I thank the management of Sarojini Naidu Vanita Pharmacy Mahavidyalaya who are conducting this, who are facilitating, funding this particular faculty development program and having invited me. So welcome to the era of quality as a culture, especially from past decade. The quality importance has raised a lot. So I begin with a little bit of historical aspects, not very historical like 50 years or anything like that. Just let's go back to 1 decade back. In 2011, 15 people were lewd, used, and discarded. So a lot of unethical drug trial got exposed. It's in Nalgonda district. It's very much in our state. After this particular thing was exposed, it has gone to the extent of Supreme Court demanding regulations of clinical trials to be strict. And court asked the government to protect the vulnerable people from poorly run trials. The question is, who are vulnerable? And what do you mean by poorly run? Vulnerable people are all the subjects who are poor, who may be mentally retarded, or who doesn't have much knowledge of decision making, whose decision making is affected. Poorly run is something which doesn't comply with regulations, which doesn't comply with good clinical practices. Yeah, in education also, from past 1 decade, we have seen tremendous development of regulations, and the faculty has become the cornerstone. 1 decade back, faculty does not have so much of importance as we have today. Okay, so one faculty, if is not there in a college, the intake of B Farm, Farm D or M Farm may get decreased. So vulnerable people, faculty are protected by the pharmacy profession regulations. In the same way, in 2013, Supreme Court ordered and government has promulgated lot of regulations to control the clinical trials. The control was too excessive, and in 2013, literally 60 to 70% of clinical trials got vanished from Indian scenario. So there is a corrective step. In 2019, central government introduced the Sugam portal, and again, facilitation of industry problems and all. So from 2019 Sugam portal, new drug clinical trial rules, NDCT rules, it has replaced, it's not addendum, it is replacement of the previously existing rules and regulations of clinical trials. It happened in 2019. So all the books before 2019, one has to check about its validity. Yeah, global clinical trial market, it's about $60 billion right now, and poised to become almost $85 billion in by 2030. And Indian share is just $2 billion in last 2 years back, and it is poised to reach up to $4 billion by 2030. So what does that mean? Right now, the place where we are, and after 2030, or say another 7 to 8 years, the market is almost getting 80% bigger. You can imagine the tremendous growth opportunities which are present in clinical trial market. In next decade, the market is going to be doubled. It's a very big growth potential clinical trial market has. So it's a potential is more in the market. So one should bother more about, learning about clinical trials, learning about quality assurance, learning about regulations, all that. Well, quality assurance and quality control, frequently they are used. Then what is the difference? Precisely speaking, quality assurance includes quality control. You can see here, quality control is a subset of quality assurance. Okay. So quality control is checking the defects, like whether case report forms are correctly written or wrongly written, whether informed consent form is present or not present, whether ethical issues are addressed or not addressed, whether any fraudulent activity taken place or not, whether documentation is properly done or not done, whether GCP is complied or not complied, whether approved protocol is followed or not followed. This is quality control. Okay. It's like whether the tablet is passing disintegration test or dissolution test or hardness test. That is what is quality control. Then quality assurance mean all the procedures which are done to minimize the defects. Okay. So failure of the process to be prevented, quality assurance has lot of steps involved. So in academics, if you take whether student is passing or failing is a quality control. And whether quality assurance in academics means whether what sort of FDPs you are doing. If you train the faculty, the student improvement can be better. Okay. By providing some air conditioners, conducive academic atmosphere, if you provide, the possibility of defects, students failing will decrease. So that is what is quality assurance steps. The conferences and FDPs and taking better faculty, all these are steps to minimize failures of the students. So that is quality assurance. So let me repeat. Quality assurance means quality control plus all the efforts to reduce the defects in the clinical trials is quality assurance. So clinical trials mean what? They are systematic studies in human beings to find out the safety and efficacy of new drugs. So because humans are involved, because unapproved drug is involved, because the new drug's safety and efficacy is unknown, because of all these reasons, the clinical trial market is very vibrant, complex, and the concerns are compliance with GCP, compliance with approved protocol is a challenge. Maintaining the quality assurance in the trials is a challenge. Not the least is the ethical issues involved in clinical trials is very, very important. So clinical trial market is pretty complex because of these three issues: ethical issues, quality issues, and compliance issues. So when you know the issues, you can find out ways of addressing it. So I think this is a very important slide in the lecture. Identifying the challenges is of paramount importance in academics also. You have to identify where do we lag? What are the things we can do to get a proper quality student out? Then we can make it perfect. You need to know who are the decision makers in clinical trials, who have some say, who have some role to play in the decision making process of the clinical trials. They are called as stakeholders. So sponsors, investigators, study participants, regulatory authorities, ethics committees, all these are the stakeholders. Here we'll go one by one. Even the subjects are the stakeholders. So sponsor is a person who initiates the clinical trial, who funds the clinical trial, who takes management of clinical trials. For example, Sun Pharma, Torrent, Pfizer, these companies who run the clinical trials of a new molecule, they are the sponsors. Lot of expectation, lot of concern is there for the sponsor. Investigator. The person who initiates is sponsor. The person who conducts the clinical trial is investigator. And investigator responsibilities are, instead of all these items, I'll tell it in just 2 lines. One line is protection of safety, welfare, and rights of the participant. The other responsibility of investigator is the research interest, the kind of data he generates, the credibility of the data that he generates, that is second item. So investigator is a person who conducts the trial, dual responsibility. One is responsibility towards the subject participants. Second is responsibility about the quality data that he generates, records, documents, reports. That is investigator. So I think in a couple of minutes, I feel that I made it clear what is investigator. He is a doctor with MD qualification and he is supported by sub-investigators, and clinical research coordinators. Frequently, they are termed as CRC. CRC means clinical research coordinator, who is a team member of this clinical investigator. Okay. Then ethics committee is another stakeholder. Ethics committee is a committee which safeguards participant safety, welfare, and rights. Slight difference is there in meaning: safety, welfare, and rights of the participant. This is a traditional role, but in the new regulatory environment, there is an expanded role of IRB. IRB is US terminology, Institutional Review Board. Ethics Committee is Indian terminology. Expanded role is ethics committees are responsible for monitoring, not just in the beginning, but in a continuing basis throughout the clinical trial conduct. Ethics committee has to keep a watch, and they are prosecutable. Earlier, it was not there, but in the changed role, they are answerable to the court of law. They are prosecutable, and they have some decision in deciding the compensation of the participant in case of some events like death or partial or full disabilities during the trial. In clinical trials, majority of the decisions that ethics committee takes or investigator takes or a company takes, they are based on whether the risk is low, whether the risk is medium, whether the risk is high. And definition of what is low risk, what is medium risk, what is high risk. It is a very subjective one, but this particular slide gives a convincing demarcation between low, medium, and high. Another person who has some decision-making role, stakeholder, is monitor. Monitor is a representative of the pharma company who will be going to all clinical trial sites to oversee, to check whether the doctors are doing trials as per protocol, as per GCP, as per SOP, as per regulations. So, sponsoring company is legally responsible. If doctor does a mistake, company will be responsible to answer it. So, sponsor has to check what the doctor is doing. How can a sponsor who are based in Mumbai or Delhi, how can they monitor hundreds of sites across the country? Answer is through these monitors who will be going to the sites and who will be going and checking each and every activity of the investigator. It is not like, doctor does some 100 files and monitor will go and check randomly some 20 files or 30 files. No. He is expected to check each and every activity that the doctor does will be checked by the monitor. So, monitor is like our students with 4-5 years of experience. And investigator is somebody like our, management who will be around 50 years, 60 years, very senior doctor. And sponsor will be like our, pharma companies like Sun Pharma, Pfizer. So how does this, our student with 4-5 years experience, how he balances this highly experienced investigator team in a very big hospital like KIMS, etc. And the other side, he has to report to pharma company head. Generally, they report to the project leader. They report to the project leader of a particular clinical trial project. So that's why this cartoon is really an apt one. Really, it's very difficult to balance this. So how to balance this? It is only through knowledge of good clinical practice, knowledge of protocol. What is the difference between good clinical practice and protocol? Anybody who can volunteer? I will tell about academic example. If I have to tell the GCP and protocol, as a faculty of, say, pharmacology, your subject knowledge of diabetes pathology and pharmacology of anti-diabetics, that is your protocol. When you go to the class, you will teach about diabetes, you will teach about drugs, pathology, and all that. That is the knowledge of protocol. Then what is GCP for a faculty going to a pharmacology class? It is like teaching methodology, the art of teaching, okay? Art of overall understanding of how to teach, okay? That is GCP. The rules and regulations of pharma education, it is common for all lectures. So that is GCP. So a pharmacology faculty has to be, should have subject knowledge and educational regulations. So clinical trial should have protocol knowledge. This monitor should have protocol knowledge and GCP knowledge. Only then he can balance the sponsor and investigator. Hope you understood. Then having said about this with example of academics, I think this slide is not required. Everyone understood the monitor role. If monitor misses the ball means a wrong document in a clinical trial process. If it is not properly catched by the monitor, then clinical trial will be a failure. So it's a difficult thing to understand maybe, especially for those who are listening to clinical trial lecture for the first time. So monitor, when he goes to hospital sites, when he verifies GCP compliance, protocol compliance, regulatory compliance, he may find some defects. Say the doctor is not taking consent from the patients, or case report forms are not proper, or SOPs are not followed. These are the defects. If monitor is missing any of these defects, then the clinical trial will be a disaster. Okay. So he has to catch the ball. And here the ball is the defects in clinical trial. And another stakeholder is CROs, contract research organizations, site management organizations, and all this. Now the question is, I hope everyone understood what is sponsor. Sponsor is like our Pfizer company, Biocon company, Sun Pharma company. Say they are in Mumbai or Bangalore or Delhi. How do they do the clinical trials across the country? They should have the logistics. Another example is, say a company is in US and they are doing trial across the globe. They need to have mandatorily a contract research organization in every country. Otherwise, they can't conduct the clinical trial. So this is contract research organization. Site management organization is another aspect, like company work is contracted out to CRO. Investigator work is contracted out to SMO. SMO concept is a relatively new one, and it is difficult to understand for the newcomers. But CRO, if you understand well, the way the projects are contracted out to CROs, investigator work can be contracted out to SMOs, site management organizations. When it is so complex, why did I write SMO in this faculty development program session? The answer is you are guiding the students, and a student with a lot of communication and management skills, they can do the startup. They can start up a site management organization by just having contact with some 4 or 5 hospitals. If they develop very good rapport with 4 or 5 hospitals, they can set up a SMO, and they can earn a lot of money and experience. Around 20% of investigator fees SMO will take and will function the trials. So reasons for outsourcing: cost is reduced, quality is improved, and speed of trials will be fast. 3 aspects: cost is reduced, trials will take place very fast, and quality will also be more. Say Reddy Labs is there. Reddy Labs expertise is in R&D, new molecule invention, production, quality control. If they are doing the clinical trial business on their own, then there will be a lot of resource, um, difficulties, and it's not their job. It's not their uh, main core thrust business area. So they will have a lot of problems. So they will contract out to some CROs like Cipra Labs or Suven Life Sciences, Quintiles, PPD, such companies. So these are various CRO companies. As all of you know, Paraxel, PPD is number one, IQVIA. Several students are going in these jobs. So, and another important difference one needs to know is auditor, inspector, and monitor. What are we discussing? Let me recapitulate. I am discussing about stakeholders or people inside a clinical trial. What are their work functions? We discussed about sponsor, investigators, monitors, ethics committee. Now remaining people are like auditors, inspectors. Okay. Now what is the difference between auditor and inspector? Auditor is like, suppose we ask our, Rambhav sir or Mallareddy sir to come our college and audit how our administration, how our academics are going on. As an external, they come and check. And if something is wrong, they will tell the principal HODs, you have to do like this, academics. Okay. They will train. And whatever mistakes they find, they tell our management. So their role is checking, doing the quality control, and also training the people inside and giving some guidance to the management how they should run academics. So inspection means like OU inspection, Usmania University inspection, or Pharmacy Council of Inspection. There, it's not the training. They check the quality and takes the regulatory action. They may decrease the intake or put a lock to the college or give an approval. So similarly in clinical trial, if US FDA comes to a clinical trial site, what do? They check GCP compliance, they check protocol compliance, SOP compliance, and if something documentation compliance, if something is not good, they terminate your clinical trial, they disapprove your product, and they have the right of administrative action, means blacklisting the investigator and even blacklisting the company. All this power they have. Then auditor in clinical trial, what does he do? Remember, when we are talking about monitoring, what did I say? Monitor will inspect, verify each and every document of the investigator. Auditor will not verify all documents. Auditor will verify random documents. Out of 100, he may verify some 10, 20. Sometimes auditor will verify by suspicion he has come. So he will verify only that document. Suppose company knows that a company like Cipla company, if they know that in Kim's hospital or NIMS hospital, informed consent violations are happening, what does Cipla do? It will send some audit team to hospital. They go to the hospital, straight away, they will go and pick up that informed consent documents and they will find the mistakes because the audit happened by suspicion. Similarly, suppose management has a idea that student feedback is not being taken, or attendance registers are not there, or examinations are not being done properly, then they will send audit team, say Mallareddy sir or say Rambhav sir or DRK sir. They will come and pick up the examination documents and take it to the management. It is by suspicion. Okay. And they will train the people. Audit by suspicion, it can be there, or routinely also audit will be there. They train the people inside and they give the report also. Whereas inspection, they don't train the people. They may give some guidelines, but main aspect in inspection is blacklisting and taking regulatory action. I hope it is clear. Inspection, audit difference. And there is slight difference, audit certificate and audit report. Audit certificate means, yes, these clinical trials in Kim's hospital are audited. They don't write findings of the audit. They just write it that audit has taken place. That's all. That is audit certificate. Class lo nanch. That is audit certificate. Audit report means it will contain all the findings. CRF is not there, informed consent violations are there. Whatever they find, they are all reported in audit report. And when a inspection to clinical trial site happens, I'm telling about regulatory perspective, okay? When a regulatory inspection happens to a clinical trial site does not need, they don't need to show audit report. They can show only audit certificate. Audit is taking place, that's all. Similarly, we may have lot of audits and all in our college. But when PCI person comes, PCI inspector comes, we don't need to show the audit report, no? In the audit report, there can be some attendance registers are not there, exam schedule is not followed. Such things can be there. All this we don't need to show to the PCI inspector. Okay. So similarly in clinical trial, audit report findings need not be shown. But one exception is there. It's very important. Suppose somebody dies in a clinical trial. Serious fraud happens in clinical trial. Suppose, then FDA team can demand the audit report. Then investigator cannot say, I don't want to show it. They have to show. When serious adverse events happen, when the inspection happens, audit report also will be insisted and you have to show compulsorily. I hope you understood difference between audit certificate, audit report. If any doubts are there, you can ask now itself. Any doubts in so far, whatever is discussed? If anybody is listening.
Sir, we'll take questions at the end of the session, sir.
నీళ్లు తాగాలి కదా అందుకని అడిగా. So types of audit I already told. Routine audits and for cause audit or by suspicion audit will be there. And also before a market approval happens, there can be some audits. Okay. So one of the very important thing in the quality assurance is good clinical practice. So good clinical practice is like GMP. So in manufacturing, everyone knows GMP. In preclinical trials, everyone knows GLP. Similarly, in clinical trials, it is GCP. So GCP is two things are there. One is protection of the rights, safety, well-being of the participants. Second aspect is the way the data is generated, the way the results are reported, the credibility, the accuracy of the data, the integrity of the trial. Integrity is a very powerful word. Integrity of the clinical trial is dependent on GCP. So I repeat, good clinical practice is two aspects. One is protection of the participant right, safety, well-being. Second is integrity, credibility, accuracy of the data. Okay. So from regulatory perspective, what do people check? One is GCP compliance. Another one is protocol compliance. GCP we have discussed. What is protocol? Protocol is nothing but it is a self-contained document. Means if a doctor has the protocol in his almara, there is no need of any other document. There is no need of any other document to run the clinical trial. Okay. So it contains aims and objectives, background, inclusion criteria, exclusion criteria, safety assessments, efficacy assessments. So it's like methodology complete. How do you manage IP, data analysis, expected side effects? All this is protocol. So a clinical trial, when it gets approved, it is the protocol which is getting approved. Okay. So from regulatory perspective, IND review is how do a regulatory body give approval for the clinical trial? The question is, how does regulatory body give approval for clinical trial? It is based on three aspects. One is quality of your formulation, your FTIR data, spectral, NMR, stability analysis, all that. So first is quality, CMC, chemistry manufacturing controls. Second is the preclinical data. Third is your protocol, which you are submitting. So these three are items for regulatory approval. NDA review means how does US FDA give approval for a product for marketing. Again, here, three aspects are there. One is quality of the product. Second is safety. So preclinical and clinical safety. Third is efficacy of the product. They do three kinds of inspections: factory inspection, safety inspection, clinical trial inspection, and they assess. Okay. So NDA is marketing approval based on three attributes: quality, safety, efficacy. So we have been discussing about regulatory perspectives. We have discussed about good clinical practice. We discussed about protocol. Now let us throw more intuition into quality assurance activities. So I think the slide is visible. Investigator meeting. What does this mean? Now we are conducting faculty development program. What is the purpose of FDP? If faculty are trained properly, they will go and train the students properly. If they train the students properly, complete academics will be going in a proper line. If proper research methodology, teaching methodology is taught to the faculty, the college will run successfully. So same thing in clinical trial. Clinical trial means, say, Sun Pharma or Pfizer is running a clinical trial globally. Let us think big. Let us think big. A trial which is running across the globe. So thousands of sites are there across the globe. And the company is in Hyderabad. Say Reddy Labs is doing a global trial, or somebody like Pfizer, Sun Pharma. They call all the investigators across the globe, they call them to Goa or Kulu Manali, or they call to Mumbai, a picnic spot, and they train the investigators. So two aspects I am saying. One is picnic spot, second is training part. Why I am talking about picnic spot is, if doctors don't come to the investigator meeting, whom will you train? So to make them come to the place of training, two things I am doing. One is giving them the training. Second thing is I am conducting it in a big 5-star hotel in which and locality is very, very famous. Suppose somebody conducts investigator meeting in Paris or any other vibrant picnic spot, then all the people across the globe will reach that point. So attendance important and training important. So investigator meetings, are they really mandatory in clinical research? Are they required? Is a big question. Even some of my students, just 2 years after they left the college, they went to Rome. I asked them why you went, how did you go to Rome and all? They said investigator meeting, I am sent to that particular. Always the main doctor will take a person who actually does the work. Suppose Usmania University conducts some curriculum change. They will call whom? They will call the principal, and they will call one fellow in the college who is a worker. Okay. So the person who really works in the college and the main person is principal. Similarly, in investigator meeting, they call the principal investigator and a key person in the clinical trial site. Now, question is it required? The answer is yes. There are two things you will train. One is good clinical practice training. Another one is protocol training. Which is more important is a question. If you don't know the good clinical practice, you know, you will be caught. Definitely, the clinical trial will be a flop. If you don't know the protocol properly, you will do some errors in the data collection, the tests that you do, inclusion criteria mistakes, informed consent mistakes, case report form entry mistakes, all this. The methodology training is important and GCP. Suppose I give another example. Suppose ciprofloxacin tablets are to be manufactured. You have to give training about the physicochemical property of ciprofloxacin, exactly how it should be tableted and evaluated. And you have to give training on GMP also. Both are important. So in investigator meetings, GCP training will be done, protocol training will be done. Both will be done. And to make all the clinical trial people to come, you do it in a big hot place. So as I said, clinical trial business is a very complicated one, complex one. It involves more people. If you go to pharma industry like Sun Pharma, Reddy Labs, you find very big machines. Okay, lot of GMP systems and all. When you go to clinical trial setup, you don't find big machines. You find big people. You find big people. I'm not talking about age. I'm talking about their subject knowledge and skills. Every MD doctor is a big shot in clinical practice, but only few doctors are good in clinical trials. Both are different. So it's a manpower intensive area. So question is, we are sending hundreds of students every year. Where are they going? They are going into this IT-enabled field where manpower requirement is very high. Even if you start 100 more colleges, absolutely no problem. The clinical trial industry can take up the load. The manpower requirement is so much. So the quality assurance requirement, naturally, it is very important element. So the study management hierarchy is this, there will be a lot of interaction between the auditors, monitors, clinical research associates with the investigators. The site environment, as I said, it is a very vibrant. When you realize the work function of the each individual stakeholder, you can realize how much interaction is required between the investigator and sponsor, between the investigator and ethics committee, among the investigator team, and sponsor is sending monitors, auditors, DSMB, IDMC to all the sites. So this interaction. So, GCP training, protocol training is the order of the day to ensure the quality assurance. There are a lot of industry FDA interaction. There are type A, type B, type C meetings possible. Again, why am I talking about this? It is, I give the example of academics. If you interact more with OU, Usmania University, if you interact more with PCI, college, then you understand them better. You can implement their strategies, rules, regulations better. Similarly, when clinical trial company, when they meet the FDA frequently, they understand the perspective of the FDA, then they can implement properly. If they keep meeting with CDSCO people, at the end of the phase 1, at the end of the phase 2, at the end of the phase 3, etc., they understand the CDSCO remarks and the strategies can be improved. So the quality assurance, it's not only investigator meeting, it's not only auditing, it's not only monitoring, it's also meeting the regulators. Okay. So these are all quality assurance steps to have the quality in place. So that's what is. And a biggest challenge in clinical trial is patient recruitment and patient retention. It's so, dear faculty, we need to train our students and improve their interpersonal skills because they are not going to handle the machines when they go into trials. They have to speak with investigator. They need to speak with monitors, auditors. They need to speak to the patients. Okay. So that particular practice needs to be, it's a key indicator of success. Again, this is a quality assurance step. If you properly manage the patients, their recruitment and retention strategies, it will lead to a proper quality control. A proper quality will be assured. If you don't follow up the patient, then definitely, patient dropouts will be there and defects will be there. They will do some wrong administration of the medicines. It's all becomes a flop. So this is very much a quality assurance measure. See, recruitment of faculty is a quality assurance step. In colleges, if you recruit correct principal, correct faculty, academics will be correct. Similarly, quality assurance in clinical trials will be perfect when you recruit a right investigator, right hospital. If you recruit the people who are known to follow the GCP, the people who are known to adhere with protocols, SOPs, the people, hospitals and doctors who are known to adhere with GCP protocol regulatory compliance. Such people you have to identify with a, what is that glass he is keeping? You have to choose and identify and magnifying glass के साथ ढूंढना है उनको. Similarly, college also should find a right principal, right faculty. Success will be automatic. So it's a very essential quality assurance step. These are all the selection criteria. After the lecture, you go through it. It's a very, very meticulously planned steps of identifying right hospital, right doctor. Yeah, another QA step is ADR reporting, identifying, and all this. I think 5 minutes more is there, or 3-4 minutes will conclude. So when a serious adverse event happens, how it should be handled. I'm not going to say how it should be handled because that lecture will be the investigator lecture. I am talking about QA perspective. You have to follow the procedures of SAE handling as per the regulatory perspective. So this is the way you have to handle the SAEs. These are existing and potential problems in the clinical trials and possible solutions. As a QA person in academics, we find out a lot of things and hold faculty meetings, train the HODs and all. Similarly, quality assurance guy in a clinical trial identifies a lot of potential problems and trains the people on possible solutions. I hope it's clear. Good documentation practice, do's, don'ts will be there. Everyone is responsible for quality. If the patient doesn't take as instructed by doctor, clinical trial will be a disaster. Am I right? If patient doesn't follow as the instructions of the investigator as per the protocol, approved protocol, then it will be a disaster. So even subjects are stakeholders and they are also responsible for quality. So the list of the things, the common defects, that common errors that people in clinical trials keep doing, okay, in informed consent process, in protocol non-adherence, these are the very frequently done mistakes among the trials. Inadequate, inaccurate records, adverse event reviews reports, eligibility criterias, they don't bother. They just recruit people just like that, don't follow procedures. These are all quality assurance steps. One has to check all these, prevent all these. So I go to the next step, that is the questions and interaction. Yeah, I invite people for any questions. Hope you
Dear,
understood.
dear delegates, if you have any questions, please, or you can write in a chat box.
ఎందుకు బాబు అందరు phones ఉన్నాయి, they can speak now. Why chat box and chatter box?
Yes, sir, no problem.
They can switch on their sound box and speak out.
Yes. Sir, one question is from my side, sir. Okay. So what are the differences in the NDA review process between the USFDA and CDSCO, sir?
Differences should not be there because
In terms of regulatory requirements,
they,
submission requirements, review process,
yeah,
or time, I think, sir.
yeah, the differences will be there. Like, FDA is more strict than CDSU. Okay. So differences mean we can talk about it. But question is, answer I want to tell is, differences should not be there because humans are humans. Participants are participants. Trials are trials, whether it is a Indian trial or US FDA trial. Regulatory perspective is 3 points: quality, safety, efficacy. Whether it is CDSCO or US FDA, they have to check these three elements perfectly. Otherwise, it's a disaster. And in NDA review, there will not be any difference because nowadays everyone is following common technical document. Common technical document is a like GMP method validation document. It is a ICH document, which is followed by US FDA, European agencies, Japan. These are three major markets in pharmacy. Indian companies also want these markets, and US companies also want Indian market. So it's like win-win situation. So whole world, they follow ICH guidelines so that their business is global. So when people are following CTD, common technical document, it is a common thing. So there should not be any differences. Procedures will vary. How they arrive at the quality, safety, efficacy may differ, but the basic things should not and are not varying. Is it clear, madam?
Yes, sir. Thank you, sir. Any more questions from delegates? Please.
Santosh, any questions? Santosh of Ranganayaka Swami College.
Yeah, sir. Yeah, I'm having a small doubt, sir.
Yeah,
Hello, sir. How are non-compliance issues can identified and how it will be resolved during clinical trials, sir?
It's like suppose PCI people come and inspect our college. How do they check this instrument is not working, that instrument is not working, attendance register may corrections are there, or faculty are less in number. So that's the way
So, yeah,
they
okay,
check.
sir.
So in clinical
Okay,
trials also, they open the trial master file,
okay, sir.
open the informed consent books. If investigator sign is not there, they just put it in dustbin.
Okay,
They
sir.
There are certain documents called as essential documents.
Mhm.
These essential documents are, when they open these essential documents, they can visualize, FDA people can visualize how the trial was conducted 3 years back.
Okay.
Like in OTT movie, crime reconstruction. In the same way, FDA can open these essential documents and they can visualize exactly how the clinical trial was conducted 3 years before. And each and every step they can verify. So it is very easy for finding the errors.
Okay, sir.
And how
Thank
to
you.
resolve? How to resolve means, if it is FDA, they close the trial and take action. If it is auditor is sent by the pharma company, no? They identify
Yeah.
the mistakes and they give the GCP training, they give the protocol training. Only two mantra: GCP
Yeah.
training, protocol training. If the trial site, like KIMS Hospital or NIMS, if they are doing intentionally something wrong, then they terminate that trial site and they take legal action against the investigator, because it's being done repeatedly, intentionally. Is it clear?
Yes, sir. Yes, sir. Thank you, sir.
Sir, my question.
Yeah.
Uh, Thank you, sir. Good information you have given. Sir, my question is, what are the parameters you will consider to quality audit for teachers?
Academics I have taken only as an example.
Okay.
Okay. Quality audit of a teacher is like GCP knowledge and protocol knowledge. In academics, subject knowledge is one, that is like protocol knowledge. Your
Okay.
subject knowledge is protocol knowledge, similar to
Okay.
protocol in clinical trial. And
Okay.
GCP knowledge is in academic environment. What does the PCI expect? What does the Usmania University expect? What does your college expect? What does your HOD expect? What does your teaching methodologies expectations are there? For placements, what is expected? In chemistry, CAD and other things, QSAR may not be there in syllabus, but useful.
Yes.
So all those things are like GCP.
Okay, sir. Thank you.
Any more questions? Dear audience, any more questions? Okay, sir.
Okay, thank you.
End the session with this. Thank you, sir. Thank you very much for your enlightening presentation on quality assurance in clinical trials, a regulatory perspective, your expertise and clear explanations of complex concepts on quality assurance, quality control, clinical trials. This is very invaluable to us, sir. Thank you once again for
Thank
your
you, madam.
sharing,
Welcome.
sharing your knowledge and the time with us, sir. Thank you, sir. With this, we end the session and we'll be back. Next session will start in a few minutes.
Thank you, madam.
Thank you, sir. Dear delegates, get ready for your final and very special session. It's going to start at 11:30 a.m. Okay, by Dr. T. Mamata, Principal Sarojini Naidu Vanitha Pharmacy Mahavidyalaya. The topic was mainly based on daily yoga routine for healthy and balanced life. So this is very important for each and every one of our life. Yoga practice is important for us also. So please join us. Dr. T. Mamata, ma'am is going to share valuable insights and practical topics on, and ma'am is going to incorporate yoga into your daily routine for a healthier and a happier life of each and every one of us. Don't miss this enthusiastic session. Please stay tuned and be ready with the mat, yoga mat.
Hello, madam. Hello.
Yes, ma'am. Yes, ma'am.
Just I want to check once these arrangements. Is it okay or not? I'll see. My video is on.
Yes, ma'am. Your video is on.
1 minute. I'll open it. Is it clear now?
Yes, ma'am. It is clear. It is clear, ma'am. Little.
It's okay. How
Yes,
about
ma'am.
audio?
Audio, little, ma'am.
Deekshita?
Maybe I think a net issue, ma'am. Now it is okay. Dr. Prashanti.
Latest ఉంటుందా?
Letters.
Camera settings లో.
Camera settings లోకి వెళ్ళాలా మ్యామ్?
Yes, ma'am.
Okay.
Good morning, everyone. Anuradha madam, can you hear my voice?
Yes, madam. Prashanti, madam. We
Okay.
can hear your voice. Yeah.
Yes, Prashanti.
Yeah, okay, madam. Thank you.
Dear all, the session is going to start by Dr. T. Mamata, Principal SNV PMV, on daily yoga routine for healthy and balanced life. I request Dr. Prashanti to introduce our guest distinguished speaker, Dr. T. Mamata.
Thank you, ma'am. Good morning, everyone. It's my pleasure to introduce our beloved Madam Dr. Mamata, Department of Pharmaceutical Quality Assurance, Professor and Principal, Sarojini Naidu Vanta Pharmacy Mahavidyalaya. She studied B.Pharm and M.Pharm from Kakatiya University, Warangal, and PhD from JNTU-H, Hyderabad. Madam is having 23 years of teaching experience, Madam is having 54 research presentations, 20 paper presentations, 2 patents, and 1 book chapter in her credit. Madam completed 8 NPTEL certification courses and stood in top 2% in NPTEL course. Madam delivered lectures in OU, JNTU-H refresher programs. And Dr. T. Mamata Madam is practicing yoga from 12 years and working as yoga teacher on honorary basis from last 5 years at Sai Yoga. Sai Yoga is founders are Dr. M. Singarao, Dr. Srimati M. Aruna. This is managed by Sri Shirdi Sai Baba Samsthan Management, which is located at Shivam Road, Hyderabad. Now over to Dr. T. Mamata. Thank you.
Dikshita, is it ready there?
Yes, ma'am. Yes, ma'am.
Thank you, Prashanti madam, for the introduction. And good morning, all. It's my pleasure to perform a yoga session on this occasion of International Faculty Development Program. And to brief about how I started this yoga. In the year 2006, we used to go to Ramakrishna Math. Means we used to go along with my daughter where she used to learn painting as well as some summer camps in the Ramakrishna Mart. And we thought, why don't we join the yoga session where the weekend yoga are going on during that time. And we joined it. And once we started doing yoga in Ramakrishna Mart, how important it is, we felt it. And one day we roamed around this 4 hours. And after that, we went for a yoga session. And along with asana, there will be a savasana. There we can relax much. And on that day, means on that spot, I experienced that performing this savasana, it's like a refreshment and nothing we did on that day. It started just like the first day. Though we have so much of physical strenuous things on that day, but the yoga made us relax, relax much, and we can start fresh on that particular day. And we decided to perform continuously, but could not do because of some regular daily routine works. And last, I joined in yoga, which is organized by Yoga Samsthan, Shivam Road, Hyderabad, by the temple management. And as Prashant said, the founders of this Yoga Samsthan are Mr. Singarao sir and is Dr. Singarao. He is a professor, retired professor from Agricultural University, and his wife, Mrs. Aruna Madam. And I started in 2012 practicing yoga. And after that, means after 3 years, we used to go early in the morning. Starts by exactly sharply 5:30 and ends by 6:30 a.m. Continued, means why I'm saying myself and my husband. Continued for years together, and in the year 2019, means I asked that I want to teach yoga because I'm in teaching profession and I'm interested to teach yoga also. I asked sir, it accepted, and 2019 onwards, I started teaching yoga also. Weekly one class I used to take, and in the during Corona period, we could not perform by going to temple and doing yoga, and we started with online sessions, and now it is getting continued in online mode. The offline also it's going on, and we have around 10 to 12 yoga teachers in that group, and the yoga teachers in that group, they are 8 years plus also. It's 5 teachers, 75 years and 80 years. They teach the yoga while performing. So that's the inspiration to me. And one day, yoga means Tara Kulkarni madam to come here to our college also. She came here and explained about the meditation as well as yoga. So they are the role models for me for this yoga practice, daily yoga practice. And this is about how I started and continuing, taking weekly once online, and I'll do on my own daily by joining online 5:30 to 6:40 p.m. And it's been 21 years. Recently we completed 21 years of annual celebrations also. And it's free of cost. All the faculty, all the yoga gurus are working. And it is supported by the temple management of Sai Yoga Shirdi Sai Baba Samsthan, Shivam Road, Hyderabad. So this is about how I started and how I started as a yoga teacher. And coming to this yoga, which I did not say the benefits of yoga. Everybody knows about that. And what we need is only nothing else. We need to perform the yoga and fresh air. Wherever the fresh air is present, we can go there with the mat and perform. And open air, especially, it is useful, and preferably in the morning with empty stomach. Whenever we want to perform, fresh air, empty stomach, and with yoga mat, that is more than enough for us. It's free of cost. And once we know some asanas, pranayama, we can do daily. And it is advised to go for minimum 5 times in a week, if possible daily, or minimum 5 times, that is better. And along with yoga, we can plan for aerobic exercises also, so that for cardiac health and other means healthy life is useful. And while performing yoga, means whatever we are going to do, it should be done with awareness. Means we have to think what we are performing, and our concentration, our focus should be on that only. So that's the main thing. And as we all know, the definition of yoga is it's a unit of mind, body, and soul. So that's why the concentration power increases, balance increases, and there are each and every asana as well as pranayama. And today we can say like generally we say Sukham Stiram Asana. Means whenever we are performing any asana, we should try to be there in that asana for a longer period of time, and stability increases, and also we feel we can feel happy in that asana. So in that way we have to proceed. Today I'm going to take 1 hour session which we do daily, which starts with preparatory exercises by sitting as well as standing position. Preparatory part where it increases the flexibility of the joints as well as increases blood circulation, and the body gets ready for, body as well as mind gets ready for the asanas. So after preparatory, we perform asanas, standing asanas, then 2 rounds or 4 rounds of Surya Namaskara, followed by sitting asanas, 1 to 6 sitting asanas, then in prone posture. Then after that, we go for supine postures. Then last Savasana, we will end with Savasana. And after the asanas, we go for generally the pranayama, 2-3 types of pranayama. After the pranayama, we go for meditation for 10 to 15 minutes. Based on the time flexibility, we perform in 10 minutes of meditation. So this is the way we start and end with. And in between the asanas that we select, the pranayama that we select means different types of pranayama that we select are different. It depends on the season as well as we can say the temperature around us. And this is not specific to any particular disease. It's not therapeutic yoga, it's for daily healthy life only. And that's what this 1 day yoga, this 1 hour yoga I am going to show you today, which starts with, generally we start with Omkara and ends with Shant Mantra. Same thing I am going to perform today, and at last, after the session, you can ask the questions to me. Then I am going to start. Generally, we will sit in the asanas. It may be Padmasana or Sukhasana. And one more thing I want to say before starting, if you are performing, don't force your body. It's to what extent that you can do, you can perform it. We should not force our body, but we should try to improve. Everybody knows that no pain, no gain. So we should try to improve day by day once you start performing this. So don't force your body. We'll start with Omkara, means in the Padmasana, or if you cannot do Padmasana, do it in Sukhasana. Comfortably, you can sit. We do 3 times Omkara. While performing Omkara, generally we will make use of Chin Mudra, where touching the tip of thumb to the tip of index finger, the 2 hands on the knees or thighs, straight, body straight, back and neck straight. Close your eyes. Then the Omkara is chanted with the letters A, U, Ma. It starts from the navel region, which will inhale fast, and while exhaling starts from the navel region, like that. Then U near the neck region, then that is in the head region. Equally, you can divide it. So I'll do 3 times. You can also do that. First inhale. You have to exhale as slowly as possible while pronouncing these letters. Third time. It ends in the head region that is called Sahasrara Chakra. So after this, generally we do chanting of Gayatri Mantra. The same now also. You can repeat after me. Om Bhuvasvaha Tatsavitarvarenyam Bhargo Devasya Dhimahi Dhiyo Yona Prachodayat. Chin on your chest. Rub your hands. Then apply the warmth on your face, neck, and hands. This is where we start. Then we start with preparatory. With the hands beside the deep region, back and neck straight, side by side, the knees as well as foot. Then now stretch the toes. Just focus my video on that. Finish. So stretch your toes, front and back. So I'm going to remove that slide. Glasses, picture and remove that slide.
It is clear, ma'am.
Then go for stretching your ankle region, means by inhaling back, exhaling front. So for 6 times like this, we are trying to activate all the nerve endings. Then go for right and left. Move right and left. We are trying to increase the flexibility of the joints. Ankle joint, knee joint, thigh joint. And together, go for rotation. Clockwise rotation. For 6 times. And opposite direction. Taking a big circle, rotate your feet. Same number of times. Relax. Then knees up and down. 10 to 12 times. Go for alternate knees. Up and down. Relax. So while performing the yoga, there are relaxing postures also. It's called as Chitradandasana. Like this, you are placing your palms on the asana and suspend your neck, close your eyes and relax. So there are in between relaxing postures. While relaxing, we have to focus on our breath. Yes. And we perform one exercise, that is Udanda Namanasana. And these two together. One. So go to Munisirisha madam and ask her to remove that slide. We will continue the preparatory. Perform Udanda Namanasana. This for legs together. Then hands at the chest region. Then inhale while exhaling. Twisting left. Then forehead on the asana. Come to center. Then again right side. We are trying to twist upper part of the body. So continue for 4 more times. So I will do for 1 more time. Like this. Also right side. So twisting. We are doing some stretching and twisting to the body. Then we continue with preparatory while standing position. If you stand and we will do Tadasana. Comfortable distance, the feet. And take your hands, interlock on the head. While inhaling, stretch your hands and legs. Stand on the toes. And while exhaling, down. Continue 4 more times. So I'll do 1 or 2 times. Just I want to show the asanas, preparatory exercises. This is Tadasana. We are trying to stretch all the muscles. Then widen your legs. Hands 180 degrees. Inhale while exhaling. Twist your body to the left. Maximum possible extent. Go to center. Then right. We'll continue. Fastly. Right and left. We are trying to twist our upper part of the body. And slow down and release your hands. And next is you can do side bending also. Again, hands on 180 degrees. Go for left side hand up. Come to center. Then right side. See that left side stretching, right side compression. And you can continue. 4 or 6 times. This is side bending. Relax. You can go for forward backward bending. Bending your legs. While inhaling, take your hands and head back. And while exhaling, hands and head together down on the asana. Continue. Inhale, exhale. Inhale, exhale. For 10 rounds like that. Then we'll go for knee rotation. Keep your hands on the knee joints above that. Rotate your knee joints clockwise. Just try to observe the movement of knee joints. 5 6 times. Go for opposite direction. While observing how to perform, that's what I said. Whatever you do with awareness, you have to. Same number of times. Relax. Then widen your legs. Go for hip rotation. It is left, front, right, back. 5-6 times. Just try to observe the hip joint movement. Then go for opposite direction, front, left, right, back. Try to observe the movement of hip joint and perform. Relax. Last preparatory practice, hands rotation, comfortable distance, repeat it. Then fingers together. While inhaling, take up from front, and while exhaling, complete the circle. Continue rotations. 10 rotation, 10, 12 rotations generally we do. Taking a big circle. Then after that, go for opposite direction, anti-clockwise, same number of times. Take the big circle, so it releases the frozen shoulders, flexibility increases, blood circulation increases, and body and mind getting ready for the asanas. Relax. While relaxing, close your eyes. This is called relaxing in Shitala Tadasana. While relaxing, we'll try to normalize our breath. We will start with asanas. Till now, we completed the preparatory while sitting in standing position, and we'll perform the standing asanas. First, we'll start with Trikonasana. 1.5 feet distance. Repeat. Right hand 180 degrees. Palms towards sky. While inhaling, take up. Upper arm touches the ear, right ear, and stretch. Right hand up, left hand down, opposite direction. Then twist your left foot. Put the foot in single line, 90 degrees to each other. While exhaling, bend to left side. Don't change the position of right hand. Only bend to the left side. Take your left hand onto the ankle, on the feet, or on the asana. What direction it is possible. So this is the final posture of Trikonasana, and you can stay in that posture to whatever extent it is possible. Minimum 30 seconds we do generally. Then come up while inhaling. Adjust your feet. Then again stretch opposite direction. Right hand up, left hand down. While exhaling, 180 degrees. Palm towards asana. Relax slowly. This is also awareness. So one side we completed in a similar way. Left side. While inhaling, take your left hand up 180 degrees. Palms towards sky. Then raise reach. While inhaling, stretch. Left hand up, right hand down.
Examiner states so exhibition always
Then twist your right leg while exhaling. Bend to right side. Right hand on the foot, on the ankle, on the foot, or on the asana. So try to observe left side stretching, right side compression, slowly side bending. Don't bend forward, side bending. Stay for 30 seconds. Okay, I'm not performing up to 30. Just I want to show all the asanas. Okay, while inhaling, come up. Adjust your right foot. Then stretch again. Left hand up, right hand down. While exhaling, 180 degrees. Palms towards asana. release slowly. So this is the way the asana is to perform stepwise while focusing on the breath as well as the body. How to perform. One more asana I will show you, that is called balancing asana. Generally useful in the daily life also. It prevents falling down as well as injuries. We do Santulna asana. Comfortable distance, the feet, balancing on the left foot. Hold your right foot, hold it at the ankle with right hand. Then take your left hand up while inhaling, touching the left ear, knees side by side, right foot touching the right heel, touching the right hip, body straight, eyes open, focus on one point in your front. Stay in the posture for 30 seconds. Try to balance. And slowly release your hand, release your right leg, release your left hand. In asana, you perform two sides in a similar way. So left side, balancing on the right foot, hold your left, back at the knee, knees side by side. While inhaling, take the right hand up, straight, eyes open, focus on the point. This side by side, stretch your right hand, stay in the posture. So this is Santulanasana. We are trying to balance, and this increases our concentration power. And whenever we are doing these balancing asana, we have to auto-suggest ourselves. Okay, while exhaling, release left hand, left foot, left leg, then right hand. So there are many different balancing asana. One asana I have shown. Then we'll perform Surya Namaskara 2 rounds. Okay. We'll do 2 rounds of Surya Namaskara. Each round of Surya Namaskara with 12 postures. What direction possible, we'll try to do. Try to improve day by day. We'll stand on the asana such that 1/3 towards up and 2/3 leave it at the back. V shape, feet, then Namaskar Mudra, thumbs touching the chest, rest of the 4 fingers towards outside. Generally we do in the morning, let's say invoke the sun god. Like chanting of one mantra, you can repeat after me. Is Om Mitraya Namaha. While inhaling, take the hands up, like 11 number. Again inhaling, take the hands and head back. That is Ardha Chakrasana. Then, as possible to you, and touching the ears, don't separate it. This is Ardha Chakrasana. Every posture, you have to synchronize with our breath. While exhaling, hands and head together. Bend forward. And hands beside the feet. One Pada Hastasana. While exhaling, you can place your forehead on the knees. 5. Don't bend at the knees. This is Pada Hastasana. Next posture. While inhaling, take the right leg back. Ashtasanchalanasana. 15 fingers in one line. Look up. Horse in a gallop posture. While exhaling, take the left leg back. Chatrangasana. Where taking the support of our palm and toes, head to toe. This is Chaturangasana. Then go back, Ashtanga Namanasana, come to Shashangasana first. While exhaling, with your chest on the asana, chin up, squat, Ashtanga Namanasana. While inhaling, take the posture of Bhujangasana. Cobra posture. Your knees can be on the asana or above the asana. While exhaling, take the support of hands and feet. Come to inverted V posture, Parvatasana. 2 feet together. This is called Parvatasana. Then it will be the repeated postures. While inhaling, take the right leg front. Again, 15 fingers in one line. Look up. While exhaling, left leg forward. Padahastasana. While inhaling, hands and head together. Then Ardha Chakrasana. Above the Navi posture, naval posture. Bend back. Then straighten the body. This Namaskar Mudra. And release the hands while touching the chakras. This is one round of Surya Namaskara, which involves 12 postures. So whenever we perform Surya Namaskara, it will be even number. First round is over, second round, where you have to take left leg back. Instead of right leg, you have to take left leg back. Then we perform with the mantras. For each mantra will take one position. 12 mantras, each one position. You can repeat after me the mantras while performing. Take the position. Namaskar Mudra. Om Vedraya Namaha. Om Ravaye Namaha. Om Suryaya Namaha. Om Manave Namaha. Om Sadaya Namaha. Om Pushne Namaha. Om Hiranyagarbhaya Namaha. Om Marichaye Namaha. Om Adityaya Namaha. Om Savitre Namaha. Om Akaya Namaha. Om Bhaskaraya Namo Namaha. Generally perform short shavasana. We are not doing shavasana. Just relax while sitting. We perform 2 rounds of Surya Namaskara. That is 24 postures. And in each posture, we will try to stimulate the energy centers or chakras in our body. So that's the main thing we do in Surya Namaskara. Relax. Generally, there will be a little bit faster breathing. Try to normalize, observing, relax. Then we'll go for sitting asanas. We will do Vajrasana. It's a simple posture. Anybody can perform it. Just fold your legs, touching the big toes, each other. Then sit such that spine is erect. This back and neck straight. So while folding the legs at knees, you are going for Vajrasana. So the asana is named such that if you sit for a longer time, your body becomes like a diamond. You become strong. Generally in this asana, the digestion process is improved if you sit for a minimum of 10 minutes. And this is the only asana which we can do at any time of the day, preferably after having meals, to improve the digestion process. Other asanas with empty stomach, only this asana after having meals also we can do. So whenever you get free time, try to sit in this Vajrasana. We will go for another asana, Ustrasana, camel posture. Go for stepwise. Just move down. Standing on the knees. Place your hands at the back. Take the support with the help of your hands. Thumbs touching last vertebra and the other fingers other side supporting. While inhaling, bend back. Go to the capital C posture. Then slowly release your right hand. Hold the right leg at the ankle or the heel. Whatever it is possible, then release the left hand. Hold it with the heel or ankle. Then it will be like capital D. This is camel posture. Stay in the posture. Enjoy the posture. That's what I said. Sukham Stiram Asana. Okay, I'm not doing longer time. Slowly release, come to the C posture. Slowly straighten your body. Release the asana. Then rest in Vajrasana. So each step we are performing with awareness. See that we are not thinking any other matters, outside matters. Only we need to focus on whatever we are doing. Focus on breath, focus on body. That's all. Whenever we do, generally the back bending asanas, we have to perform forward bending. You do Sasankasana. Stretch your hands. While inhaling, take the hands up. Hands and head together, little bit back. While exhaling, forward bending. Only because please don't lift your hip. Place your hands and head on the asana. Hold it on the asana. Take back your hands. Rapid posture, Sasankasana. Shallow breathing. We are pressing our internal organs, lungs. Stress management asana. If you can do this for longer time, stress is released. While releasing, with your hands. While inhaling, hands and head together. Release your hands. Relax. This is rapid posture. So most of the asanas, you can see that they are like this here. All the asanas are taken from the nature, that is animal postures are taken. Then we'll go for one of the relaxing asana, that is Makarasana. Lie down on the asana, prone posture, one palm over the other, widen your legs, and chin or forehead on the hands. Relax. This is crocodile posture where all the body parts are touching the asana. Relax, relax. So whenever you feel physically stressed, you can go for this asana. Blood pressure is also going to be normalized. Every muscle and cell is getting relaxed. In the prone posture, we perform one asana, that is Bhujangasana. Sit together. Hands near the hip region. Palm on the asana, palms on the asana. While inhaling, head up, chest up, body up, perpendicular to the hands. And this knees can be also a little bit up, far on the asana, whatever is possible. It's like cobra posture. Imagine it's a possible stretch. This is one of the best asana to relieve from back pain. So the waist region or back region makes it very minor problems. They are going to be relieved by this asana. Lungs are getting expanded. Release. Then slowly release the asana while exhaling. We'll go for one more relaxing asana. It's called Shitlasana left. I'll show you Shitlasana right. Where right hand, right leg straight, left hand, left leg. Fold it. Right cheek on the asana. Close your eyes. Relax. This is also one of the relaxing asana which lowers blood pressure. Means whenever you feel high blood pressure, you can relax in this asana. You roll out your back. You go for supine asanas. Roll out your back. We perform two asanas in this posture. One is Pavanamuktasana, next is Set Bandhasana. Pavanamuktasana, Parsha Pavanamuktasana means we will do with right leg, left leg, and with the both legs. First with right leg, hold the right leg, hold below the right knee, inhale, hold the breath, and press it with the knee. While exhaling, touch your forehead or nose to the knees. This is asana where the gastric problems are relieved. While exhaling, while inhaling, head back, exhaling, right leg, release right leg. This is also stepwise. Then we will go with left leg, hold it, hold the left leg below the knee, inhale, hold the breath, and press it to the chest. While exhaling, touch your forehead or nose to the left knee. While inhaling, head back, exhaling, left leg release. I'm not doing for longer time, just I want to show the asanas. Then with both the legs, Purna Pavanamuktasana. Hold. Then hold below the knees. Inhale, hold the breath, press it. Try to observe. Blood is rushing towards face. It's like facial. You do not go to the beauty parlor. Blood circulation is rushing towards face. Then while exhaling, head up. You want to perform the dynamism to the asana? You can do. You go for falling like this. Okay. Then while inhaling, head back. Exhaling, stretch your legs. Relax. This is Purnapawanukasana. Generally, the gastric problems like constipation is prevented or relieved from that. Best asana for that. Then we will go for Setubandhasana. It's also called as bridge posture. Mainly helpful for back pain, where spinal cord will be taking rest. I'll show you. Hold the legs, heels touching the hips. Then the support of it as well as the hands. Lift your body up part by part. That lower back, middle back, upper back. Then take the support of hands. Place it like a pillar to the bridge. Now, body is looking like a bridge. This is Setubandhasana. Relax. Supram Siram Asana. Enjoy the asana. Slowly release the asana. Part by part down the asana. Stretch your legs. Relax. Relax. Do Shavasana, Sampurna Shavasana. This asana. Let's shake all the parts, legs, hands, head, right and left. Adjust so that all the body parts on the asana. Put in your legs. Face inside, toes outside. Side hands, palms towards sky. Hold your fingers to little extent. Close your eyes. Relax. Relax. Totally, your mind is focused on the body only. That's what you have to do. So, the mind goes somewhere else, but you have to get it back. Thoughts, you have to get it back and focus on the body. It's relax. Relax and relax. Just try to observe with your third eye. Try to observe on toes to knees, both legs. So, relax. Observing your third eye and relaxing. A similar way, observe on these two navel regions. Observe and relax. And now we will do neck region. Try to observe internal organs also. Relax. Relax. Try to observe from neck to organ chakra, that is coordination, body, facial muscles sound in relaxed condition. Nothing should be in stiff position, everything is in relaxed condition. Relax. Away from Agna Chakra to crown region. All the sides of the crown region. Try to observe and relax. This is what we call as Panchatattva. To observe part by part and relaxing. Don't think that we are losing energy by performing the yoga. Generally, we gain energy. Relax. This is the way we do the Shavasana. The athletes While lying on the down, we perform Tadasana. Hands, thighs, take it back, legs together. Inhale, hold the breath, then stretch, hands up, legs down. Tadasana. Opposite direction, to possible extent. Stretch, stretch. While exhaling, relax. Turn to right. Your hand, right hand like a pillow. Then left leg on the right leg, right hand on the left thigh, left hand on the left thigh. The support of left hand. Come to sitting posture. So we completed all the asanas. Then go with pranayama. What's the time? We do Vastrika pranayama. Then we'll go with Nadi Shuddhi and Brahmari pranayama. So Vastrika with hands movement will do. This is with forced inhalation and exhalation. One step I will show you where we will take Adho Mudra at chest level. While inhaling, you take the hands up, unfold your fingers. While exhaling, down and again Adho Mudra. So this we will perform in 3 speeds. First with slow speed, then medium, then come to high speed. Again go to medium as well as slow speed, and each speed we will perform 10 times. So we'll start with slow speed. You can also perform. So inhale, exhale. Inhale, exhale. Inhale, exhale. 10 times, continue. Try to concentrate on inhalation and exhalation with synchronization of hands movement. We are trying to increase the capacity of our lungs, clearing all the channels. Then come to medium speed. Inhale, exhale. Inhale, exhale. Inhale, exhale. Continue. Go with high speed. Inhale, exhale. Inhale, exhale. Inhale, exhale. Continue. Medium speed. Inhale, exhale. Inhale, exhale. Continue. Slow speed. Inhale, exhale. Inhale, exhale. Inhale, exhale. Continue. Relax. Relax. This is Vastrika pranayama. Means in pranayama, little bit heavy pranayama only, but is one of the useful pranayama. So with it's also called as alternate nostril breathing. Anybody can perform this at any time, only a little bit empty stomach, fresh air around. most beneficial pranayama this is. I'll tell you the stepwise process. Left hand on chin mudra, and right hand Nasika mudra. Nasika mudra we are folding the index finger and middle finger, stretching all other fingers. This is Nasika mudra. So first you have to close our right nostril using our right thumb. Close means don't press it, just touch it. Then exhale through left nostril. Then after that, inhale through left. Then close it with right hand, ring finger, while exhaling through right nostril. This is off round. Then inhale through right, exhale through left. It's one round of Nadi Shuddhi. Alternate nostril. Inhale through left, exhale through right. Inhale through right, exhale through left. So while practicing, we also include holding of the breath. After inhalation, hold the breath. After exhalation, hold the breath. It's called internal kumbhaka and external kumbhaka. Purakam, Rechakam, then internal kumbhaka, external kumbhaka. We'll do 2 rounds. Just I want to show you how to perform Nadi Shuddhi Pranayama. And here also the back and neck straight. While inhaling, to fill all our lobes of the lungs, and while exhaling also means hold the breath for a few seconds. While exhaling, it should be as slow as possible. Empty the lungs. Feel that you are emptying the lungs. After that exhalation, hold the breath for 2 seconds. Then go for second round. It is to be continued. Noise and totally focused on the breath only. Perform with awareness, then only will have full benefits. So while practicing for months and years together, the breath rate comes down. When you see the animals and human beings also, once the breath rate is more means when your means anxiety or anger increases the breath rate and automatically decreases the lifespan. If you take the example of tortoise, it's having more lifespan, the breath rate is very less. So like that, by practicing, we have to try to decrease the breath rate, automatically increases the lifespan. So the way benefits and calms the mind and can increase concentration or focus increases, clarity increases. So many benefits of this. The last pranayama, Brahmari pranayama, is also called as deep breath. We'll do with application of Shanmukha mudra, that is placing thumbs on the ears, index and middle finger on the eyes, then ring finger on the upper lips, little finger on the lower lips, and tip of the touches the upper palate. We'll inhale, while exhaling, we'll make a humming sound like bee. Just perform 5 to 10 rounds daily. So it is also helpful in making the mind calm, and generally it improves or enhances the action of parasympathetic nervous system. In that way, it is much helpful. We'll do 2 rounds. Apply Shanmukha mudra. Inhale. While exhaling, humming sound. While practicing, you can experience it. It's practiced correctly, then you can feel the vibration in the head region. Just close your eyes and observe that after performing the Brahmari. Last, we'll do the meditation and close it. So, coincidentally, today is World Meditation Day. Just yesterday only I got to know that. Only perform 10 minutes. So while doing meditation, you can sit comfortable posture, hands in chin mudra, place it on thighs or knees, and you can adapt any method like focus on breath, focus on inhalation and exhalation, or you can chant Omkara silently in your mind, or you can go for japa, that is chanting the mantra either outside or inside, in a rhythmic fashion like Om Namah Shivaya, Hare Rama, Hare Krishna, you can chant. So any of the single method or try to be on that single thought for a few minutes, closing your eyes, also back and neck straight only. But in between we'll get other thoughts. Just try to remove that and come back to your single thought only. So that by practice you get more on that and you'll be out of it's in your own world. Forget about outside what is happening. A few minutes. So this is the way we have to perform meditation. Concentrate on or focus on singing whatever it is. We came to ending the session. So generally we do with Three Omkara and Shant Mantra. So whatever we started with Three Omkara, in the way we do that. That is Auma with exhalation, synchronizing with exhalation. So go for inhalation. Of inhale. Shant Mantra, you repeat after me. Om Shanti 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 Loka Samasta Sukhino Bhavantu Sarve Bhavantu Sukhinaः Sarve Santu Niramayah Sarve Bhadrani Pashyantu Kashchit Dukkha Bhaveत् Om Shanti Shanti Shanti. Chin on the chest, rub your hands, on face, neck, hands. Stretch your legs. Loosen your legs. Still now you are in the asana, sitting asana. So then get back blood circulation to the legs. This completes 1 hour session, 1 hour 10-minute session actually. So on behalf of this, I'll say thanks to all my yoga gurus, Dr. Singarao sir, Anna madam, Karakula Karni, Pratibha Hegde madam, and Venkateshwar Reddy sir. We are one group performing daily, and daily 1 hour for our physical and mental health. Most important, where we can be away from the medicines. I can say that, means I am proud to say that I am not using any medicine as of now. That's good. It's because of yoga and because we need to interact with many people daily during the work life or domestic life and handle everything. May feel stress some cases, and at the end of the day, maybe under stress, but once I perform this 1 hour yoga in the morning, I'm relieved of everything. So this is the way getting utilization of this asana and pranayama in my daily life, so that my professional as well as work life is balanced, I can say. Thank you, one and all. Thank you.
Thank you, Dr. T. Mamata. Thank you, ma'am. My sincere thanks to Dr. T. Mamata, ma'am, for conducting this yoga session. And every step you take here will help you, and you may grow stronger, and you may become more balanced and more mindful. Remember to carry the peace and mindfulness you have cultivated here into your daily life until your next time. Take care, all of you. Thank you for conducting this session once again. And my sincere thanks to participants also for joining this session. Thank you all. Over to Anuradha, madam.
Yeah, madam. Thank you, madam, Dr. T. Mamata, for your.
Madam, your voice is not audible.
Not audible.
Anuradha, madam.
Yes.
Please check. You may be in unmute. Mute.
Yes, yes. Am I audible now? Prashanti? Prashanti?
Yeah. Yeah, ma'am. I can hear you. I can hear you. 1 minute. Some questions are there in chat box.
No questions.
Madam. Amita, madam. Madam, yeah. Thank you, madam. My question is, yoga, can we reduce hernia? Madam, itlandi.
Can we reduce umbilical hernia, madam? Question is, madam, with yoga, can we
Yes,
reduce
yes, sir.
umbilical
Yes,
hernia?
One more question
specific
asked
type,
again, madam.
specific type of yoga means it should be done under the guidance only. Actually, as of now, I can't say this particular asanas or this particular pranayama is useful for your particular disease or something, because generally it should be done with the advice only. Means as of now, I cannot say. Means we have different types. In the therapeutic yoga is also one of the type. Okay, so only what I have shown today is it's for regular daily yoga and for regular health only. It's not specific to any disease. So there are certain asanas and pranayama specifically meant for specific type of diseases that can be performed.
Okay, thank you, ma'am.
Madam, one more question is there from Harita Sunil. Madam. Madam, one more question from Harita Sunil. That's wonderful, madam. And if it was in the early morning, we would have practice along, but a great shaking something.
What is that question?
Best appreciation.
Okay. Preferably early morning is good. Means after 4 or 4:30, 5:30. And if you don't get time, we can perform in the evening also. But little bit means empty stomach after having meals. There should be a gap of 1.5 hour to 2 hours. Then that's better. And generally means you can go to the grounds or any fresh air area. If you're performing in the room, closed room, so open area is preferred always. Yes. Okay, we can close this session. You can continue with the closing remarks, Anuradha madam.
Okay. Okay, madam.
Assessment forms are shared.
Yes, ma'am. Already shared. Forms are shared.
Closing remarks.
Madam, review of verdict. Madam,
Yes.
shall I continue, ma'am?
Yeah, continue, madam.
Okay. With the review of program? Shall I
Yes.
continue with the review of
Yes.
Okay. 1 minute, madam.
Madam, all the delegates can switch on their videos, madam, for taking some screenshots.
Actually, yesterday we have taken the feedback. A few have responded with the introduction. So today also, please, if anybody want to say something, put on the video and we can speak on this because we are going to close this e-FDP program today itself. So if you want to say something, put on the video and say.
Delegates, you can share your valuable feedback on our program. Any suggestions also welcome. Please respond. Please switch on your video. Kiran sir.
I know anybody is from.
Kiran sir. Kiran Vema. Dr. Kiran Vema. You are not audible, sir. Sir, not audible, sir.
Am I audible now, ma'am?
Yes, sir. Please.
Ma'am, this is Dr. Vema Kiran from Mohan Babu University. I have been following the FDP right from day one, but literally to speak, I could not follow many because of other works. I want to congratulate the team because the wide selection of topics were there. Even the speakers were very good. Actually, the word good is because they enlightened very much about the topics. And I congratulate the whole team of Sarojini Mahila Vishwavidyalaya. And I congratulate all. Thank you, ma'am.
Thank you, sir. Thank you for your feedback. From which institute you say? Mohan Babu Educational Institute.
Mohan Babu University, ma'am, Tirupati.
From Andhra Pradesh, yeah, Tirupati.
Anybody outside of Telangana, please respond. Other states. Yesterday, one responded who is from Assam Downtown University. And registrations means from Madhya Pradesh, Tripura, Just we want to know whether they are there in the session or not. Okay, Ganesh sir, Sri Ranganayaka College of Pharmacy. How the sessions are going on, sir? Okay, Ganesh sir. So, sir, Ganesh sir, are you there? Okay, anybody want to speak, please?
Yeah, Harita madam from St. Paul's. She participated actively in all sessions, madam. She's
Yes,
my
ma'am.
ASCOM guide.
Okay.
Yeah.
Yeah, ma'am. Actually, I've shared my feedback already. That's why I'm not unmuting myself to share the feedback again. However, all the sessions I have attended successfully. Maybe today morning session I could not attend for the whole session. And Mamta Madam's final, it was like a grand closing for the FDP by giving the very well yoga session. And actually, if it was in the morning at 6 o'clock, we would have practiced along with you, madam. But definitely, we will take these suggestions and we do the practice. Thank you so much for conducting such a wonderful, and I really appreciate all the efforts that has been taken by the entire team. And congratulations for the entire team, madam. Thank you so much.
Thank you, madam.
Anyone from any other institution? Vaishali madam?
Yes, ma'am. Good afternoon, ma'am. The last session is very good, ma'am. And ma'am did so great, ma'am. They also she made good things, ma'am. Means and how much impact yoga on our health, ma'am.
100%, madam.
Yes, ma'am. It's good, ma'am. Actually,
It's like it's more than 100%.
Thank you, ma'am.
Because it's not physical, right? We focus with our mind.
Yes, ma'am. It is mentally also relief, ma'am.
Everything about yoga, we cannot miss. It's few thousands years back started in India only. Means we have the credit of that, and we started with International Yoga Day with the support of means by the initiation of Narendra Modi ji, and they all by experience written so many books on yoga. It's only part of it. Okay,
But you did it great, ma'am.
but so much to learn. It's beyond our pharmacy. Means everything has got some with scientific reasons and scientific proofs. As a pharmacist, we can understand. Means it stimulates each and every system of our body. I said during Nadi Shuddhi Pranayama or Brahmari, it stimulates parasympathetic nervous system.
Yes,
Like that, everything we can go with
activated.
scientific. Thank you.
Thank you, ma'am.
Brother, madam?
Yes, madam. Okay, we are at the end of the sessions. All the sessions.
Just let me speak for 2 minutes.
Yes, madam.
We are at the end of this closing of International Faculty Development Program. So it's a proud moment to all of us, especially the Department of Pharmaceutical Quality Assurance and Saroj Naidu Vanta Pharmacy Mahavidyalaya. And on behalf of this, I say thanks. Means as a principal, I want to say thanks, always thankful to the management, starting with our chairman, sir, Dr. B. Prabhat Shankar, sir, and our honorary secretary, sir, Dr. B. Should be Hanmant Rao, sir, and Sandhya Deepala, madam, Chandradeep Singh, sir, and also vice chairman, sir, Chala Prasanna Garu, and also the GB members who are so active as well as encouraged all these activities, co-curricular activities, and whenever you ask any activity to perform, they say yes every time. So this is the way the encouragement we are getting from our management, and that's why we could perform without any problem. Means though we have some curricular works, academic works are going on, and I can say thanks to all the team members of pharmaceutical quality assurance who did very well, coordinated very well, and here HOD of pharmaceutical quality assurance, Dr. Anuradha madam, and Dr. K. Vinuta, and Dr. Shivajyoti madam, and yesterday Prashant madam became doctorate. I congratulate on behalf of this once again, Prashanti madam, and Mrs. Munisirisha, who supported a lot to us, our department, with means technical support as well as doing all these things. Though she is from chemistry department, and missed one of our colleagues. She is on leave. She is Mrs. Sretasri. Anyone I missed? Yeah. And yeah, Dr. Shivajyoti madam is going to take care about the certificates of participants. She'll do that. And yeah, this is a good teamwork, we can say. And in future also, we'll be having department-wise activities. We'll continue the same. Thank you one and all the participants, those who registered from Telangana as well as outside Telangana. We say thanks, and actively participated, actively interacted with the speakers also. And whenever you want any speaker's email ID or phone number, we are ready to share with you, because all the speakers, those who contacted, we contacted them, and they are ready to help you all. They said okay to share the email IDs as well as phone numbers also. Thank you one and all. Over to Anuradha madam.
Thank you, madam. Good afternoon, dear delegates. I'm Dr. S. Anuradha, Head of the Department Pharmaceutical Quality Assurance. I would like to express sincere thanks to everyone who made this grand success. Okay, who has contributed to the success of this event. I would like to give the overview of our program. It's a faculty development program. The theme of our program is pharma quality assurance and regulatory affairs, a collaborative approach. It's a 6-day faculty development program, and for this program 125 members registered from various parts of our India. So we invited 12 guest lectures by eminent national and international academicians and industry professionals. So in this program, we started our program. We started the day 1 on 16th at 9:30 a.m. With the inaugural ceremony by welcome address. Dr. T. Mamata Madam gave a welcome address, and the next opening remarks by Dr. N. Srinivas, Professor and Director of S.N. Vanitha Pharmacy Mahavidyalaya. After that, we started our sessions at 10 a.m. By the inaugural speaker. First speaker, Dr. M.V. Narendra Kumar Taluri. He's a director, technical person, and he gave a session on OECD principles of good laboratory practice, and we thank sir. And then next session 2 started at 11:30 a.m. and with a distinguished speaker, Dr. Srikanth Nama is a managing director, QARA at SS Innovations International, Gurgaon. And sir gave a topic on emerging innovations and breakthrough in the pharmaceutical industry. Sir gave a wonderful session on novel topics, advances and the latest topics. It's enlightened all the delegates. Next session, day 2 started at 8 a.m. Session 3 started at 8 a.m. by distinguished speaker Dr. R. Bhaskar Rao, Senior Director, Regulatory Affairs, Clinical Therapeutics from USA. Sir gave a topic on clinical IND and NDA submissions, regulatory dimensions. Next session was at 11:30 a.m. by distinguished speaker Dr. Mr. J. Naidu, Management Consultant and Career Guidance Coach from Pune. So sir gave a topic on joy of teaching. It's very important for all the teaching faculty. So he gave a valuable information to all the faculty. Then on day 3 we had a session at 8 a.m. Session 5 started at 8 a.m. by distinguished speaker Mrs. T. Rajani, Senior Business Validation Lead, Mark from USA on computer system validation. Then next session 6 was at 11:30 a.m. by distinguished speaker Dr. Aparna Vema. She is a Senior Director, Immunocure, on computational chemistry. She is from working in Hyderabad. Next, she gave a topic on AI in pharmaceutical R&D, a game changer for drug discovery and beyond. And day 4 session started by Mr. V. Narendra Kumar. He is the AGM, Anta Pharmaceuticals, Hyderabad. His topic is on implementing a robust quality management system in the pharmaceutical industry. Then section 8 on 19th day by distinguished speaker Dr. Linga Banot. He is a senior scientist from CSIR ISCT Hyderabad. Sir, topic was on enriching excellence through process capability and sustainability in cascade process with enzymes. Then day 5 session that started at 10 a.m. by distinguished speaker Dr. M. Vidyavati. She is a professor from Sri Padmavati Mahila University, Mahila Visavidyalayam Tirupati, Andhra Pradesh. So madam gave a topic on DOE for optimization in formulation development. The next session on day 5, session 10 by distinguished speaker Mr. Mohammad Rahim, a corporate quality lead, Tobacco Pharmaceuticals from Saudi Arabia. So sir gave a presentation on ensuring excellence through process capability and quality systems. The next today's session by our director Dr. N. Srinivas on quality assurance in clinical trials, regulatory practice. And last session, it is a very special session on health. This is very important for each and every one of us. One should adapt these methods, procedures to build our health and maintain our health. Okay, by Dr. T. Mamata. Topic was on daily yoga routine for health and balanced life. Okay, these sessions, today's sessions mainly focused on impact topics in the field of pharmaceutical science, offering insights into the regulatory compliance and clinical trials, and promoting the health through yoga for the balanced life. Both the sessions which are conducted today that I'm talking about. Next, the key highlights of our program is in these sessions, 12 sessions, we covered critical topics in the pharmaceutical industry, including regulatory frameworks for ensuring compliance, innovations in pharmaceutical R&D, mainly focusing on AI-driven drug discovery. This is very important for each and every faculty, how drug is discovered. From that only we can go for formulations and all the other activities. So innovation topics that we learned with this session, with this e-FDP program. Next, quality management system and the importance in ensuring the industry standards. Then optimization techniques for pharmaceutical formulation developments. These are the key topics of our sessions. So these topics mainly provided valuable insights into the latest trends, regulatory aspects and technical achievements in the pharmaceutical industry, and exciting and excellent. This is a very important platform and excellent platform for the knowledge exchange and collaboration among the pharmacy professionals. So this is about the review on of our faculty development program. Now, I would like to thank all the persons, our management who actively shared their guided us to make this event grand success. I would like to thank. It's a great honor to extend our heartfelt gratitude to all those who contributed to the success of this event. So I would like to thank everyone. Firstly, we would like to extend my sincere thanks to our Chief Patron, Dr. B. Prabhashankar, who is a chairman of Sarojini Naidu Ananta Pharmacy Mahavidyalaya, Managing Director and CEO of Lead Pharma Private Limited, Hyderabad, and the President of Indian Pharmaceutical Association, Telangana State Branch. for his unwavering support and encouragement to conduct such programs in our department and in our college. Sir always encourages us to conduct programs like this. So we also wish to thank our patron Sri Challa Prasanna, Vice Chairman of our SNVPMV, for his valuable guidance and contributions for our college. Next, a special thanks to our co-patron, Sri B. Hanmantrao Garu, Honorary Secretary, SNVPMV, for his contributions and leadership and visionary for the development and growth of our institution and developments in our college. We would like to acknowledge Srimati Sandhya Deepala, Honorary Treasurer, and Sri Chandrajit Singh, Joint Secretary of SNVPMV, for their valuable support and coordination. Always they used to give us valuable suggestions for each and every moment, each and every event which we conduct in our college. I thank our management and our convener, Dr. T. Mamata, Professor and Principal of Sarojini Naidu Vanitha Pharmacy Mahavidyalaya. She played a vital role in organizing this event. We extend our deepest appreciation for her dedication and hard work to make this event a grand success. We also extend our gratitude to our co-convener, Dr. Sri N. Srinivas, Professor and Director, Sarojini Naidu Vanitha Pharmacy Mahavidyalaya, for his valuable support, encouragement. Always, sir, guidance is very important for all the departments in our college to conduct such programs. We thank you, sir. Our coordinators, Dr. B. Shivajyoti, Associate Professor, Dr. K. Vinuta, Associate Professor, Dr. R. Prashanti, Assistant Professor. On this platform, I would like to congratulate Dr. R. Prashanti. Day before yesterday only, she awarded PhD. So one more faculty member got a doctoral degree in our department. On this platform, I would like to congratulate her. Congratulations, Dr. Prashanti. Next, I thank Mrs. Moni Sirisha. She gave a support to the quality assurance department. Always, she's a technical person. She always very very active, and she will take care of all the technical aspects, and she supported support is very important for the success of this program. Thank you, Mani Sirisha. She is assistant professor of Sarojini Naidu and the Pharmacy Mahavidyalaya.
Thank you, ma'am. Thank you.
Lastly, we thank all the HODs of various departments of our college, and teaching faculty and supporting staff, and most importantly, our esteemed delegates. So with your support only, with your active participation only, our faculty development program is became a grand success. Thank you one and all, okay, for your active participation, contribution, making this event a remarkable success. So we truly grateful for each and everyone for their effort and making this memorable and impactful experience. Okay, this is the first time. For the first time, we are conducting e-faculty development program from our department. We could succeed with all your support. Thank you one and all for your support.
2 minutes, ma'am. Vinuta madam want to talk. 2 minutes.
Yeah, please, Vinita.
Hello everyone. I'm Dr. Vinuta, working as an associate professor in the Department of Pharmaceutical Quality Assurance. First of all, I would like all the participants your hearing to this e-FDP, which is conducted by SNDPMB. Thank you one and all. With your encouragement, we look forward to conduct to do some more online FDPs. Thank you one and all.
So 1 minute, in our department, we are a team of 6 faculty members, including our principal madam. One more faculty is missing. She is Mrs. Sweta Shri. She is on leave, but she is also actively participated and gave her inputs to make this event. Thank you. Thank you. Next, Prashanti, please carry on. Prashanti, you want to say something?
Thank you all for participating in this and conducting this. Thanks for our management, director and principal, our HOD, Anuradha madam, everyone for conducting this e-FDP. Thank you all for participating and encouraging us. Thank you, ma'am.
Shivajyoti madam.
Good afternoon, one and all. And I would like to extend my thanks to management first who supported us in to conduct all this e-FDP and selection of all speakers, everything. And all the delegates who participated actively and make this program grand success. And in future also we'll conduct more and more events like this with your cooperation. Thank you, one and all.
Mrs. Moni Sirisha.
Yeah, good afternoon, one and all. Yeah, first of all, I would like to thank the participants because whoever we sent the invitation, they have registered. Thank you so much. Without your registrations, this cannot be get success. And I thank management, then principal madam, director sir, and HOD of QA department, Anuradha madam, for giving the opportunity to participate in this e-FDP program. And in future, we thought we'll collaborate and work like this together. And thanks to all the audience for your patience listening. And you will be receiving your certificates very soon. If any technical issues or anything you want to give, we are going to send a feedback link, overall feedback link for you through WhatsApp. You can fill it and you can give your open suggestions so that we can rectify any problem arised during this 1 week session. Thanks a lot. Thank you, ma'am. Thank you.
Thank you, Sirisha. Actually, for your, I would like to thank you on this platform for your tireless efforts and organizing and ensuring the smooth flow of this event. And you are there for each and every moment for the technical support. Thank you, Sirisha.
Thank you, ma'am. It's my pleasure, ma'am. It's my pleasure. Thank you. Thank you so much.
Thank you, one and all. So you can, if you are interested, you can stay back in this. We can continue in a like a WhatsApp group. If anything, any event is going on in our college, we will post these events or other things. Delegates, if anyone wants to say anything, feel free to say. We'll post the certificates to your registered mail ID. Anything to say? Thank you, sir. Thank you, Ashok, sir. For your support and encouragement.
Madam, one more thing I would like to say, ma'am. Any delegate due to their classwork or some busy schedule, if you might have not attended any session, you can go through our YouTube links, because every day as YouTube links was posted in the YouTube channel of Sarojini Naidu Vanitha Pharmacy College. We'll post our YouTube channel in the group. You can check it out and you can go through with the sessions. All the 6 days sessions are there. Thank you.
Sir is Ashok Kumar, sir, our R&D advisor.
Good afternoon, sir. Good afternoon. Good afternoon. Yes, sir.
Yes, ma'am, you can close the session.
Okay, thank you, one and all. Dr. Anuradha signing off.